Good morning, everyone. My name is Quinea, and today I will be presenting on pioneering new drugs for new drug targets for inhibiting c-Myc expression. And I did this while I was working in Dr. Hurley's lab over the summer. So I'm just going to fast forward to my methods because that's my final. Okay, so this is a diagram of how I'm going to break down my methods, and I do have a little bit of results that I'm going to present within. I don't have all of them today, but I will have them for the final presentation. So what my project was I was discovering drugs to inhibit c-Myc expression, which is it's a cancer gene that is found in, it's overexpressed in over 80% of cancers. So it's usually genes are expressed in normal healthy cells, but in certain cancers, c-Myc is expressed tenfold. It's expressed a lot more. So we're trying to repress it. So what I initially started off with was a 3,000 compound library screen. And how I did this was we used it with FRET, which is a fluorescent resonance energy transfer. And how this works is you have a specific gene or DNA sequence that you're working with, and you can insert two different markers that express different things. So you'll have a fluorophore on one end, which emits a wavelength of light, and then you have a quencher, which acts like a black hole, and it sort of sucks in this light. So how you do it is your donor fluorescent, so you have this excitation, which kind of excites the molecule, and it'll emit a light. And then you have an acceptor, and they're supposed to be in proximity within each other. So you have your fluorophore and your quencher. And if your drug is targeting your gene of interest, and if it's stabilizing it, it will pull these structures together, and you have your fluorophore and your quencher close together. And what that does is it won't emit a wavelength, so it'll have a decrease or an increase. Or actually, it'll have a decrease because you're quenching the light. But if your drug compound is interacting with your gene of interest in a different way, if it's destabilizing it, it'll pull the two figures apart, and your quencher isn't within close proximity to your fluorophore, so you have an emittance, and the black hole can't suck up the light. And this is just a nice fancy way of showing a wavelength. And what you want is you want them to be within close proximity so that you have this, so that you can get a read off in a machine, and we use a spectropolometer. This is some results that I have. This is the actual sequence, and you can see the fluorophore is on one end, and you have the quencher, and the I motif again is a C-rich or a cytosine-rich structure, and you can see you have your runs of cytosines, and that's what forms the actual I motif. And what we were looking for is we were looking for decreases or increases in fluorescence that were over 50%. So this shows that the drug is directly interacting with the I motif, and it's decreasing the fluorescence, I guess. And these are just the fancy ones. And this one right here in particular, we actually progressed into further studies. So after we screened about 3,000 compounds, which took pretty much a semester of work, we then investigated how they were specifically interacting. And we did this with circular dichroism, which is also short for CD spectroscopy. And how CD works is it takes linearly polarized light, so it shines a light through your sample, but it takes it circular. So it's almost like you're taking an image of someone's hand from the left side in a counterclockwise position and the right side. So you're taking it directly from two different sides, and you don't get the direct image of your hand, you get a shadow effect. And this shadow we can analyze, and we can sort of like use, we get the raw data and we'll analyze it, and we'll put it into a nice little format where we can say, okay, is this drug directly inhibiting or promoting this little spectra that we get? And there's a certain signature that we can get from it, and it's called a spectra. And the molar ellipticity, so these are the actual results. What is shown up here is that with water and DMSO is what our drug compounds are actually saturated in. They're just in the solution. It's short for dimethyl sulfoxide, yeah. And So you can see at about 8 molar equivalents, which means that there's about a ratio of 8 drug compounds to 1 I motif in the solution. You can see that it's already decreasing molar lipticity. And molar lipticity is just a concentration of I motif species in the solution. So with drug, this is actually decreasing the amount of I motif present, which will, later on show that it's decreasing c-Myc expression. And then this is just a stoichiometric binding curve, which is another thing that we can measure. It's just more molar equivalents, how much drug is actually interacting with our I motif species. And then right over here we have a thermal melt, which when you have no drug impacted with water edema, so you get the stability, you can track the stability of the I motif. And it usually melts at about 40 degrees Celsius, but when you add drug, you can see that it's decreasing or increasing slightly. No, I think it's decreasing, and then it starts to increase at 15 equivalents, but you're actually decreasing the stability of the compound. So the drug is, what we're thinking that is happening is the drug is interacting with the I motif, and it's sort of putting it into this other structure, secondary structure that we don't really know. And then that's that's the actual compound, what it looks like. And so after we go through and we run these titrations on all these different compounds, then we can actually look at target specificity. And this is through potency on the target. So we do this in cell work. And I don't think I have any data on that yet. After we look at the specificity of the target, then we do what's called an MTS assay and mRNA isolation. So we actually take our drug into cancer cells, we grow them up, and we sort of look at, is the cell able to penetrate, is the drug able to penetrate the cell? Is it able to interact, surpass all these different biological restrictions, and is it able to get into the nucleus and impact? So this is an MTS assay, and what it is to just, we grow up cells in a 96 well format, and we drug the cells at different concentrations, so you can see that the gradient is going this way. And this just is an explanation of the process that occurs. So what we do is we add our drug to our cells, and then we read it on a computer, and the colors, the MTS buffer that we add will change color. So if the cells are living, it'll change into like this dark amber color, and then they'll send out a signal, and then we'll be able to read, okay, this many cells survived with this concentration of drug. And if we if we see a lighter color after we have added the MTS buffer, then we know, okay, the cell is, this drug is cytotoxic, it's killing many of the cells. And then we know at what dose we get an IC50, which is an inhibitory concentration for 50% of the cells to be living and 50% of the cells to be dead. And this is an example of another drug that we found, and you can just see this is a nonlinear regression curve, and at 50% right here, this is the concentration of drug that actually killed more than 50% of the cells. And then we also use QRT PCR, and this is actually to look at, is our gene of interest being repressed? And how this works is you have a single-stranded DNA. So what we do is we take our drug with our cells. Then we do mRNA isolation. So we isolate the cells that we want, or we isolate the RNA that we want. So we'll have a single-stranded DNA, and this is just an example of double-stranded. And you have your you have a TACMan probe, which has a fluorophore and a quencher, and it'll come in and it'll bind to your DNA sequence. And what PCR actually does is a primer comes in and it starts transcribing or translating the DNA, so it'll start pairing up with all the other base pairs. And if you do have enough RNA in there to be messaged or to be paired with, it'll actually eat up this little probe right here. And when it eats up this probe, you get the fluorescent, the fluorophore and the quencher separate, and so the quencher can't get rid of the light so it fluoresces, and then you can read it off on a little machine. And that's kind of what we use. And then characterization, this is for modes of, I don't remember what MOA stands for, but it's we can do further assays. We can do a scratch assay, and we can look more at protein working, because what a gene does is it signals for certain proteins to be made. So if we can actually progress a drug into this stage, and that's what my further studies will be, if we can progress a drug into that stage, then we can probably even take it further and maybe into clinical trials or, small animals. So that's pretty much the flow through of what I've been doing.